a food microbiological lecture by Assistant Professor Marwa Al-Khafaji for the 4th stage of Biology Department in the College of Science, University of Baghdad. Our lecture number 5, talking about the factors affecting microbial spoilage of the food. There are many factors affecting microbial spoilage of the food, most of them related to food, so we call it intrinsic factors, while others surrounding the food, extrinsic factors. We'll talking about some of them. They are affecting the type of spoilage and the fast of spoilage. Number 1, the number and the kind of microorganism in the food. Generally, when the number of microorganisms on the food surface is high, the spoilage will occur faster. As we see here in this figure, the y-axis log number of bacteria, while the x-axis is the days of slim appearance on meat surface. So, when the meat piece is loaded with microorganisms with high microbial load, so it will spoil faster than when it's with low microbial load. This is regarding the number of microorganisms. But it's not always true. This is not always true because in spite of high number of microorganisms on the food surface or on the food contaminated with microorganisms, these microorganisms must have the ability to consume that food. So, in the protein food, the spoiling microorganism must be of the group of proteolytic microorganism that can consume protein food. And in the fatty food, these microorganisms must be lipolytic microorganisms to have the ability to spoil fatty food. So, not only the number of microorganisms affect food spoilage, also the microbial type of the microorganisms that spoil that food. They must have the ability to hydrolyze such food. So, proteolytic in protein food and lipolytic in fatty food. The second factor is the chemical composition of the food. We will classify foods into 3 categories: protein foods, fatty foods, and sugar foods. Regarding protein foods, the main spoilage of the protein food is called putrefaction. So, protein food with a true proteolytic microorganism will be converted to polypeptides, and these polypeptides with a proteolytic microorganism will be converted to amino acids, which will be break down to butyrate compounds, gases, amines, and organic acids. There are 3 main pathways of amino acid breakdown: deamination by deaminase enzyme and decarboxylation by decarboxylase enzyme, and the third pathway is about tryptophan amino acid only, where it be converted to scatol and indole by the bacteria Escherichia coli. The second category of foods are sugar foods. The common spoilage of them is fermentation. The type of fermentation determined according to the sugar content. So, we will classify sugar foods into 4 categories or classes according to sugar content. Foods with low sugar food content, foods with low sugar content like meat. The type of fermentation is a bacterial oxidation. The type of spoilage and low sugar food, just like meat, will be bacterial oxidation, and the product is organic acid with gases. The second class of sugar food is medium sugar food, like milk. The type of spoilage is organic acid fermentation, as lactic acid and organic acids gases will be formed. The third class of sugar foods is high sugar food, like juice. The type of spoilage is alcoholic fermentation by yeasts, so alcohols will be formed. The fourth class of sugar foods is very high sugar food, like honey. It will be spoiled but a very slow alcoholic fermentation by osmophilic yeast, and a little amount of alcohol will be formed. It's called yeasty honey. This yeasty honey is due to improper storing in humidity, so it will be a very slow alcoholic fermentation by osmophilic yeast only. The fatty foods, the main spoilage of them is rancidity. There are two main types of rancidity. Number one, oxidative chemical rancidity, that is caused for all types of fats when it exposed to non-microbial rancidity during the storage in light and sun. So it is non-microbial rancidity due to storage in light and sun. This rancidity occurs as a result of an autochemical reaction. An autochemical reaction due to storage in light and the sun, causing oxidative chemical rancidity of fats and fatty foods. This is autochemical reaction. In the fat produced rancid fatty acids, ketones, and aldehydes. The second type of rancidity in fatty foods is the microbial type, called microbial hydrolytic rancidity, occurred by the activity of microorganisms of the type lipolytic microbes. So the lipid bilipase enzyme from the lipolytic microorganisms will be converted to fatty acids and glycerol, and then to ketones and rancid aldehydes. So this is microbial hydrolytic rancidity. The first one was autochemical reaction, not by the microbes. The third factor is the food pH. Foods with a neutral pH are suitable for growth of most of bacteria and fungi, so it will be spoiled faster. But this table will show the relationship between food pH and food spoilage. We will see in the first column the food type, then the food pH, and the main spoilage microorganisms. So, alkaline food with pH of 8 to 10, just like egg white of a pH 9.6. The microorganism which can spoil such food is alkalophilic microorganisms, as some strains of Pseudomonas and Proteus. Neutral foods with 6 to 7 pH, most of bacteria will spoil it. Low acid food, just like fruits and vegetables, the spoiling microorganisms are acidophilic bacteria like lactobacillus. Medium acid foods, the medium acid just like a fruit, juice, or fermented milk. Most yeasts and lactobacillus can spoil such medium acid foods of 4 to 5 pH. 3 to 4 pH of the acid foods like pickles or juice will be spoiled by lactobacillus or acidovector. Foods with high acidity of 2 to 3 pH, like lemon juice, which is 2 pH value, rarely spoiled if occurs by fungi. So the high acid juice with 2 to 3 pH is rarely spoiled if occurred is by fungi, not by bacteria. So this table showed us how is the food pH affects the microbial spoilage and the type of the microorganism which can spoil such food. The other factor is food moisture. The food moisture is very necessary. So water activity or the active water is the important part of the food moisture that is necessary for the growth of bacteria and enzymatic activity in the food. The table show the microorganism which could cause spoilage to foods with the lowest active water value. So the fresh meat and the fresh cheese with the active water of 0.91 will be spoiled by bacteria. Salted meat and salted cheese with AW of 0.88 will be spoiled by yeast. Dry fruits and grains of active water with a value of 0.8 will be spoiled by molds, while salted foods with an active water of 0.75 will be spoiled by halophilic bacteria. The grains and the dry fruits with an active water of 60.65 will be spoiled by xerophilic molds only. Sugar food, jams or honey will be with an active water of 0.6 will be spoiled by osmophilic yeasts. Milk powder or cornflakes of active water less than 0.5, they will not be spoiled by microorganisms, except when we open the cover and let the active water to be higher than 0.5, then it will be spoiled either by osmophilic yeasts or xerophilic molds, and this is by inappropriate store conditions. There are other factors less important, but they are affecting the microbial spoilage of the food. I'll list some of them only, which are biological structure of the food, just like when the food has a hard cover, so it will be more resistant to microbial spoilage. The presence of antimicrobial substances in the food, like polyphenols in tea, it will inhibit microbial growth, so it's seldom to be spoiled. Aerobic and anaerobic conditions due to some practices with the food, like meat mincing, so the bacteria on the surface will be in the bottom and vice versa. These factors and other factors will be affect the microbial spoilage of the food. Thank you for listening. This is an abbreviated lecture of the food spoilage and the microbial the factors affecting the microbial spoilage of the food. It is abbreviated to be suitable for the time of my students. Thank you so much for your listening. I'm available for any question.